Chapter 8, Energy and Enzymes, an Introduction to Metabolism. In this chapter, we're going to see how enzymes use energy to drive the chemistry of life. So we're going to talk about chemical reactions and discover how we can categorize them as both spontaneous and non-spontaneous. We'll look to see how enzymes play a role in actually accelerating chemical reactions and what factors actually affect enzymes and how enzymes work in metabolic pathways. Because the next two chapters will be specifically on metabolic pathways, that is photosynthesis and cellular respiration. Okay, energy and enzymes. Well, notice we're going to talk a lot about words that begin with the letter E, like energy, and they're all associated with energy. And the amazing thing about energy is that I can't pull something out of my pocket and say, hey, this is what energy looks like. It's usually associated with something else. And I think maybe for this reason, it makes it sort of a difficult topic to talk about. So please make sure that you study and look at this chapter very carefully. So it says here, energy and enzymes make cellular activity possible. So we need energy for chemical reactions in the cell, and we also need it for movement and other activities in the cell. So activities are constantly changing in the cell, and they're typically mediated by enzymes, and these reactions need energy. So metabolic pathways are a series of chemical reactions that will either build up or break down molecules. Okay, so there's two types of energy, and we've discussed this before, so this is just a reminder. The two types are kinetic energy and potential energy. Kinetic energy, like kinesiology, is energy of motion, kinesiology of motion. And we can measure this using thermal energy, and it's the energy of molecules moving around. And I always think in my mind about boiling water, all those molecules, H2O molecules, like jumping around, all those bubbles in a pan of boiling water. Then the second type of energy is potential energy, and that one's sort of harder to visualize, but I always think about the Franklin Mountains. You know, I'm either at the bottom of the Franklin Mountains or I'm at the top. When I'm at the top, it takes a lot of energy to get up there, whether I'm walking, biking, or even in a car, because it's going to take a lot of gas to get up to the top of the mountain. And when I get to the top, then I have a lot of potential energy. Energy that's stored in the position, the high position that I'm in. The other way of thinking of potential energy, it's the energy that's stored in the bonds of a molecule, and that is also referred to as chemical energy, energy stored in chemical bonds. And that's what we'll be talking mainly about in these next two chapters. So energy can be transformed from one type to another. In other words, we can convert from kinetic energy to potential and vice versa. In this example here, this is a transformation. We see a red ball at the very beginning or at the very top of a high waterfall. It has high potential energy, but as it moves down, that energy is converted to kinetic energy. Energy is lost through kinetic energy. And then finally, the ball hits the surface at the bottom, and when it does, it makes a loud noise. There's mechanical energy and also heat. So all of that energy is released in those forms. So here we see the transfer of energy moving from potential energy to actually kinetic energy and other forms of energy. So where is the energy in a molecule? It's in the bonds. So there's potential energy in the covalent bonds. So remember, this is potential energy. The electrons are being shared by different atoms. So it says how much energy is there? Well, it's relative to where it's located with regard to the nucleus of each of the atoms. So potential energy depends on the position of the shared electrons. So remember, we have different types of covalent bonds. They're polar and nonpolar. So if the electrons are shared equally, then it's a nonpolar covalent bond. And when they're shared equally, there's a lot of high potential energy. But if the bonds shift to the side, then they have lower potential energy. In figure 8.2, you can see that carbon and hydrogen share electrons equally. It says long weakest bonds, but notice here, if you look at the arrow down at the bottom of the figure, you see it has high potential energy. And as the electrons are shared unequally, because both nitrogen and oxygen are electronegative, you see that there's a loss of potential energy. Chemical reactions. Chemical reactions involve energy transformations. So chemical reactions can either release energy or they need to acquire energy for the reaction to occur. So remember, energy is in the bonds, and when we think of a chemical reaction, isn't it true that we're simply thinking of actually a reaction to me is breaking bonds and forming new ones? So there's a change in energy that is transformed to thermal energy or to light. So one of the things that we need to remember and that you will need to know for the exam is the first law of thermodynamics. And you've already heard this one before. The first law of thermodynamics is that energy is conserved. It cannot be created nor destroyed. It can only be transferred or transformed. Now, this is a new term, enthalpy. It's another word that begins with the letter E. And when we're going to see this in an equation in just a little bit, and it's going to be designated by the letter H, and it's the total energy of a molecule. So let's think about this. If most of the energy is the potential energy in a molecule is in the bonds, then the more bonds a molecule has, the more total energy it has. So it makes sense that large molecules have a lot of total energy or enthalpy, and small molecules with very few covalent bonds have less total energy. So keep in mind, total energy is the inherent energy in an atom like Brownian motion. In addition to the bonds, the potential energy that I was just talking about. So we can have a change in total energy, or this is primarily based on the difference in potential energy, and this can be caused by either an exothermic or endothermic reaction. So we take the words apart. Exo means exit and thermic is heat. That's a form of, remember, kinetic energy. So exothermic, there's a release of heat. And when that happens, delta H is negative. So when you release heat, then the molecule has less energy and therefore it's negative. So products have less potential energy than the reactants. In an endothermic reaction, we're building something. So endo means going in. It needs energy. Heat is going in. So it's taken up during the reaction. Then delta H is positive, and products have higher potential energy. Does that make sense? Yes, it does. Because if you're building something, then it's going to require more atoms, and the molecule is a lot larger, and it has a lot more bonds. So therefore, that energy was used to form bonds and to create a larger molecule. Okay, so you remember entropy from chapter 2. Entropy is designated with the letter S, and it accounts for the amount of disorder. So remember how I think of disorder? I think about it in very simple terms. Large molecules are very ordered, and so entropy is low. Small molecules show a lot of disorder. So notice I'm referring order and disorder to actually the size of the molecules, the size of the molecules. So entropy has to do with small molecules and disorder. So it says here, when products of chemical reactions become less ordered than reactant molecules, then entropy increases. So when you're going from a big molecule to a small molecule, that means that there's an increase in disorder. Does that make sense? Yes, it does, because large molecules indicate order and small molecules indicate disorder. This is the second law of thermodynamics, states that total entropy is always increasing. Figure 8.3 shows actually the creation of disorder. When you see all the billiard balls together, it reminds me of a large ordered molecule. But when entropy is increased, notice all the balls are bouncing off each other. Instead of being clustered together, they become individual balls. And in this case, this reminds me of small molecules indicating greater disorder. So the other thing, all of this is leading up to this important formula that you will be tested on. And so the question is how is it that we can track energy transformations, or how do we know which way energy is going? So there's this formula, Gibbs free energy, and you can imagine that it was developed by Dr. Gibbs. So it tells us whether we have a spontaneous or non-spontaneous reaction. So the one thing that's new in this equation before we get started is the figure delta. And delta here looks like a triangle. So this triangle means a change. So when you see delta G, that means a change in G, or a change in free energy. So what is the equation? Delta G is equal to delta H minus T times delta S. So if we take a look at a change of Gibbs free energy, what does it really amount to? Delta H is total energy of a molecule minus disorder. And notice that disorder is multiplied by T. So that means as temperature increases in degrees Kelvin, that actually we see that there's greater disorder. So when delta G is less than 0, that means we have greater disorder. And when delta G is positive, that means the molecule has more total energy. And we can see this from this relatively easy equation. Okay, so let's take a look. It says changes in Gibbs free energy equal to changes in enthalpy. Okay, so remember there's enthalpy and there's entropy. So let's take a look. It says here, chemical reactions are spontaneous when delta G is less than 0. That means delta G is negative. If delta G is negative, that means that the reaction is exergonic. That means that we're going from very large molecules and they're being broken down to small ones and they're releasing energy. Well, where was all of that energy? It was stored in the bonds and now they're broken. When delta G is greater than 0, that means that delta G is positive, and that means that the molecule now has more total energy, and it requires energy for the reaction to occur. So that means this will not be a spontaneous reaction. We only have spontaneous reactions when delta G is less than 0 and it's exergonic. So in an endergonic reaction, we need energy because we're going from a small molecule to a large one, and that energy will eventually be stored in the bonds. Then we have delta G equal to 0, and that means that the reaction is at equilibrium. Knowing whether delta G is less than 0, greater than 0, and equal to 0, and what it indicates will be important, especially on your upcoming test. Temperature and concentration affect reaction rates. The higher the temperature, the higher the amount of collisions of atoms, and therefore the higher probability that a reaction will occur. The higher the concentration, that means there's more molecules there, and there's the increase in the probability of collisions too. So the higher the concentration of molecules and the higher the concentration of temperature will increase the rate of reaction. There are two classic experiments, and actually these were done by students in chemistry, that actually prove that temperature and concentration can increase chemical reaction rates. If you look at the bar graph below in orange or the linear graph in blue, you can see that increases in temperature increases the reaction rate. Increases in the concentration of molecules also increases reaction rates. So you see the bar go up higher as you have a greater concentration. So the y-axis indicates reaction rate. So both temperature and concentration increase reaction rates. So how is it that non-spontaneous reactions can occur? So if they're non-spontaneous, how is it that we can drive them to occur? So there's this thing called energetic coupling. That means that exergonic reactions are actually coupled with endergonic reactions. And to me that makes sense, doesn't it? If we go back to the first law of thermodynamics, that energy cannot be created nor destroyed, in an exergonic reaction, where does the energy go? It has to go somewhere, and it's often transferred to a reaction that requires energy. So here, energy is not created nor destroyed, it's transferred, right? It's transferred to another reaction. So energetic coupling makes sense. I love this diagram because it shows how an exergonic reaction is coupled with an endergonic reaction. One reaction releases energy and the other reaction requires energy, and this is how they're coupled together so that you have different products. Notice an exergonic reaction has low energy products. Typically these molecules are much smaller than the reactants. In an endergonic reaction, notice that you have high energy products, and this is because the molecules are typically larger than those that were the reactants. Another thing that we need to know when we're talking about transfer of energy, as we saw in energetic coupling, is oxidation and reduction. Oxidation is a loss of electrons. Reduction is a gain in electrons. And so there's this movement of electrons that is happening in energetic coupling. In other words, one way for energy to move is that it moves with electrons. Now, it sort of seems strange that when we talk about reduction, that it is actually a gain of electrons, that it's the reduction of the charge of the electrons. Remember when we were talking about ions and how they have a charge as a superscript? It's the reduction of that charge. So oxidation means loss, reduction means gain of electrons. And the mnemonic device, and it's in your book, it's oil rig. Oil rig. Oxidation is loss, reduction is gain. Notice here, oxidation means it's an exergonic reaction. Reduction is an endergonic reaction. You can view this video clip if you look at your regular presentation, so this should be there, but I'm going to move on to the next slide. Okay, so it says here, during redox reactions, electrons are either gained or lost. So it says here, how is it? It's a change in the number of electrons in the valence shell. Remember from chapter 2, the valence shell is the outermost shell of an atom. electrons transferred as new covalent bonds formed with other atoms. So as we form bonds, we're actually storing potential energies. So either an atom is going to lose electrons or it's going to gain them. So an atom is either reduced or oxidized. I love this example, and actually we have an entire chapter dedicated to this chemical reaction. And this chemical reaction is that of actually cellular respiration. Cellular respiration is the oxidation of glucose. We're breaking down glucose in the presence of oxygen to release energy, and that's why you see all of that flame on the right-hand side of the reaction. Also notice that we're not only breaking a big, large molecule like glucose down to CO2, a one-carbon molecule, but notice that a lot of the electrons have shifted from non-polar that are shared in the middle to polar covalent bonds. And this also shows a loss of energy, and that energy has been transferred to those flames that you see as the sugar is melting in that spoon. Another thing that we need to talk about when we're talking about oxidation and reduction, since it's the movement of electrons that are carrying energy, is that we can also say that there are electron donors and electron acceptors. If you're an electron donor, then you're losing electrons. If you're an electron acceptor, you're gaining electrons. So the other thing that's moving with these electrons is oftentimes a proton. But when we talk about a proton, what are we talking about? We're actually talking about hydrogen, as you can see where I'm pointing with the arrow right here. So this proton refers to hydrogen. So we know when molecules are oxidized or reduced by tracking the hydrogen or the proton that is moving with these high energy electrons. So two famous electron carriers are flavin adenine dinucleotide and nicotinamide adenine dinucleotide. And that's a mouthful. I'm not going to ever say these again. I'm just going to refer to them as FAD and NAD. So how do I know when they're oxidized and reduced? I know that they're oxidized when it's FAD or NAD. And how do I know that they're reduced? I know that they're reduced because they have hydrogen at the end. Notice FADH2 means that it is, not only does it have protons, those hydrogens, but it also means that there are electrons. The same thing with NADH. It is the reduced form. It's carrying electrons. And that hydrogen there at the end indicates that it does. So FAD and NAD are electron carriers for cellular respiration. And I think of them as like little molecular taxis. They're picking up electrons in one place and actually moving them to another place and letting them out. So if they're picking them up, it's reduced. If they're releasing them, it is oxidized. So these electron carriers are both reduced and oxidized. FAD oxidized, FADH2 reduced. NAD oxidized form. NADH is reduced. So for those of you that are interested in chemistry, especially organic chemistry, here you can track the movement of hydrogens and electrons. For this class, all you need to know is that NAD and FAD are electron carriers, and they're moving electrons from one place to another in the cell. The other way that electrons can move is through phosphate. And the most important phosphate is that is attached to ATP, adenosine triphosphate. So what makes ATP so energy rich? It's that triphosphate tail, that TP at the end of ATP. So that's why it's the energy currency of the cell. So energy moves in two ways in the cell, It moves through electrons and it moves through phosphate. So for this reason, ATP has 3 phosphate groups, and for that reason, it has high potential energy. It has so much potential energy, I sort of liken it to dynamite. It has so much energy, but it's so unstable at the same time. And that's why our body usually produces ATP as we need it. We can't store large amounts. Wouldn't it be neat if I could like fill up my tank once a week like I do my car and just say I have enough energy for the week? But the truth is I can't. And that's why I'm constantly breathing and eating to produce ATP. So here we see ATP. This is the second way that energy moves through the cell, and it moves through phosphate groups, and you can see these phosphate groups highlighted in yellow. So ATP has 3 phosphate groups, and one of these will break off, and with that energy will travel with that phosphate group as it attaches to another molecule in energetic coupling and provide enough energy for a reaction to occur. And here you see how ATP, another visual look of ATP using the space filling model. Here you see the stick model to represent ATP. Okay, ATP reacts with water during a hydrolysis reaction. And you should remember this from chapter 2. Dehydration synthesis, and the opposite is hydrolysis. I always think of hydrolysis. Hydro meaning water, lysis meaning break. So when water comes in, it breaks off one of the phosphate groups in ATP. And when it does that, it releases a lot of energy. This makes it an exergonic reaction because it is releasing energy. It is releasing energy. So it says here 7.3 kilocalories of energy are released per mole of ATP. So here you see the reaction of how water in a hydrolysis reaction actually releases one of the phosphate groups. And remember, energy moves through phosphate groups or through electrons in the cell. So here when we break off the phosphate group, what is it releasing? 7.3 kilocalories per mole of ATP. Okay, so what do we call it when the phosphate groups move around? When one phosphate group moves from ATP to another target molecule, we call this phosphorylation. And this term is really important. You know, my best analogy is like duck, duck, goose. You know how children sit around in a circle and one child goes around and it taps on each person and goes duck, 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 duck. And then finally they touch someone and they say goose. That person has to stand up and chase someone around the circle. To me, getting up and running around is a lot of energy. So phosphorylation is like when you goose somebody and you attach a phosphate, and now all of a sudden there's a huge increase in potential energy, enough potential energy for a reaction to occur. So phosphorylation can couple exergonic and endergonic reactions. So this is one way of transferring energy. I like this diagram to some degree. I don't think it's the most perfect diagram, but it shows how exergonic on top, the exergonic reaction on top, indicated by the orange line, is coupled with an endergonic reaction. So notice at the top, we have ATP and one of the phosphate groups will be broken off, and that phosphate will connect to the reaction down below. And that movement or that transfer of phosphate with energy is called phosphorylation. And that's basically what it's showing. This is one way that endergonic and exergonic reactions are coupled together. Okay, so now we're going to start talking about enzymes. We've talked about energy, so now let's talk about enzymes. So in front of every single reaction, there's this thermodynamic barrier. And the best way I can describe it is it looks like a speed bump. If you were to draw it out, this thermodynamic barrier would look like a little speed bump. You know, that little bump in the road that makes you slow down your car so that you're not driving so fast. Another way of stating what that barrier is, we can call it activation energy. It's like the energy that you need to overcome the reaction to make it occur. So it's like a little protective barrier. Okay, so we need kinetic energy to overcome it. And remember, kinetic energy is the energy of motion. So it's like me as an old lady trying to get out of the chair. I'm so old, I'm sitting in this chair. I go 1, 2, 3, and then finally I can have enough energy to push myself out of the chair. Well, that's exactly what's happening at the cellular level, even the chemical level. There is this kinetic energy that is used to overcome this activation energy. But what does an enzyme do? An enzyme lowers this like molecular speed bump, this activation energy, and when it's lower, then I don't have to work so hard to get out of the chair. It's sort of like you when you're driving. If there's a low speed bump, you don't have to decrease the speed of your car. And then it says there's this transition state. And this transition state is the point where bonds, are weakened so that they'll break and new ones will form. So it says free energy of transition state is high. And we'll see this in future slides in the diagrams. Okay, so here we see it. So here we have the reactants A plus BC. And remember what a chemical reaction is. It's the breaking of bonds and the forming of new ones. So here you see activation energy. That's that thermodynamic barrier. That's like that little speed bump that you see in the road. But here, this speed bump is in front of every single reaction. And kinetic energy must be used to overcome the reaction. So notice at the very top, instead of seeing solid lines, you see dotted lines in between all of the reactants. Well, this transition state indicates that bonds will be broken and new ones will be formed. And then once it's done, we see that we have some new products. It's AB plus C. So I'm looking at this and I see that the free energy of the reactants is much greater than that of the products. So what is this? Is delta G negative or positive? In other words, is this a spontaneous reaction? What do you think, guys? Yeah, you're right. It is a spontaneous reaction. Delta G is negative, and so no energy was needed for this reaction. It says enzymes help reactions clear 2 hurdles. Well, what are these hurdles? They make molecules and atoms collide in a precise orientation. And it says here the other thing that they do is they lower activation energy. They lower that thermodynamic barrier. So I'm equating thermodynamic barrier with activation energy. So it's like a high jump. If you lower the bar, it's easier to jump over. If the bar is really high, then only a few can jump over the bar. So we can consider enzymes as catalysts. So what does a catalyst means? It means it speeds up the reaction. But how does it speed up the reaction? It speeds up the reaction by orienting the molecules just right and lowering the activation energy. By lowering the activation energy. Okay, so this is how we speed up reactions, or how enzymes do. And remember, all enzymes are proteins. So it says here, when reactants undergo chemical reaction, they bind to an enzyme. And now we consider them substrates. So the reactants are substrates. And we can consider this like a lock and key model. Each enzyme is very specific for each chemical reaction and the reactants, or the substrate. So substrates bind to the enzyme's active site. So imagine an enzyme is a huge macromolecule, and in a space inside, that is where the substrate or the reactants will fit, just like a key inside of a lock. And in this active site, that the molecules will be oriented in such a way that they will be able to react. And once they're inside this active site, it's sort of like Alice in Wonderland. All of a sudden, the enzyme is going to go through a change. Remember in Alice in Wonderland, when Alice puts the key into the door shrinks or becomes really large and the doorknob starts to drip? That's a change. And that's exactly what happens to enzymes. It's like science fiction. They go through this change, and this change is called a conformational change. And when they start changing their shape, actually they start stressing the bonds. They stress the bonds so they're easier to break. And this lowers the activation energy. When you stress the bonds, it lowers the activation energy. Okay, so it says here, enzymes lower the activation energy. And why do they do this? Because they weaken the bonds. They weaken the bonds that are there. Because remember, a chemical reaction is breaking bonds and forming new ones. So we have to first break bonds before we can form new ones. So enzymes decrease the activation energy. They lower it by stressing the bonds. I'm going to give you an example. One of the things is, now that I'm an old lady, is it's very hard for me to open up a jar. So what I'll do is I'll try, I'll bang it, I put the jar under hot water, and if I still can't open it, I have to ask one of my sons. And when I ask one of my sons, I go, "Oh, Mom, how come you can't open the jar?" And then they pretend that they're struggling by opening the jar. And they go, "Mom, I can't open it," and then they give it back to me. And when I test the jar to open it up, the lid just pops off. Well, what happened is they popped it open for me to make it easier. They were the enzyme. They lowered the activation energy. They made it easier for me to open up the jar, or in this case, for the reaction to occur. Okay, now there's 3 steps to how an enzyme works. There's initiation, transition, and termination. Initiation is where actually the reactants or the substrate fits into the active site of the enzyme, and they're positioned so they're oriented just right. The transition state is when the bonds are stressed through induced fit, this conformational change, just like my son helping me open the jar. And then the last phase is the reaction occurs, so the enzyme releases the products. I love this diagram here because you can see that actually enzymes are very large macromolecules because they're proteins. And remember in your book, the color green always refers to proteins. And notice here the substrates, glucose and ATP, are going to fit into the specific active site of the enzyme. And when they do, notice the enzyme is going to change shape. It's going to stress the bonds, and this is called induced fit conformational change. I love this diagram. I love figure 8.13. Notice enzymes change activation energy. They lower the little molecular speed bump, but they have no effect on delta G. Notice this is an exergonic reaction because the products have less free energy than the reactants. So here an enzyme lowered the speed bump, which is the activation energy E sub A, which represents this thermodynamic barrier. So this is how enzymes work. This is how they speed up reactions. I love this three-step process. It indicates initiation, transition, and termination. But what's neat about this diagram, it shows the lock and key type model of enzyme with substrate. And then you see that it goes through this conformational change or induced fit in step 2, the transition state. And that's why we see dotted lines between A, B, and C. And once we have our products, notice that the catalyst, the enzyme, releases the products. So it says, what are some of the limits of the rate of catalysis? Well, there are some limits to it. So this is the thing, is I'm going to tell you a story before we talk about this, and this is my story. I remember when my children were young and I was working more than full time, and I was always in such a mad rush to go home, and because I had to fix dinner because they were going to be hungry. But inevitably I would be missing something, so I'd have to rush to the grocery store to get some food, like the missing item that I needed to make for dinner. So I'd rush into the store and I'd look around and get my stuff and put it in the cart and rush to the checkout. But all of a sudden, when I get to the checkout, there's a lot of other people just like me rushing to make dinner because they're making it for a family when they go home. So as much as I want to get up and I'm thumping and I'm thumping and I'm getting anxious, what's really controlling the rate at which I can get home is not me. It's the person at the checkout counter. So the person at the checkout counter is the enzyme, and I am one of the reactants. So it doesn't matter how fast I'm going. What's really controlling the reaction is the amount of enzyme or the people that are manning the checkout. So this is how they knew that enzymes existed, because you'd figure if you have a lot of reactants or substrate, then you'd have a lot of product. But the truth is that it goes up linearly for a while, but then it curves, it settles off. So why is it that it plateaus? It goes linear and then it plateaus. And the reason is, it's because it's not just a two-way, it's not just reactants and product, it's reactants, enzymes, and product. So it goes linear and then it plateaus off, indicating that the enzyme itself has become saturated. So are there limits to an enzyme catalyzed reaction? And the answer is heck yes, it's the enzyme itself. So in this graph here you see for the x-axis substrate concentration, and for the y you see the rate of product formation. Notice if it's uncatalyzed, that means an enzyme is not involved. It's a linear reaction. But in our bodies, most reactions are catalyzed. So it's going to be like the red line. So you see that it goes up linearly for a while, but then it trails off. It plateaus off. So this means that the enzyme itself has become saturated. That means that no matter how much reactant you have, the limiting factor is the quantity of enzyme. This is called saturation kinetics. Saturation kinetics. In other words, what is saturated? The enzyme itself. It's like the people, when I go to the grocery store, I'm always saying, come on, open up more checkout lanes. Each of these people, they're doing their best, but they're still not fast enough. I want to go home. So that's what's happening at the cellular level. There's only so much enzyme, and they're already going at maximum speed. So the enzyme, or the amount of enzyme, is what's affecting the rate of the reaction. Okay, what else affects the rate of reaction? So the truth is enzymes do not act alone. They act with other molecules, and you'll need to know this for the exam. They act with cofactors, coenzymes, and prosthetic groups. And I know all of this way too well. Now as an old lady, now I care about my health. Now I don't want to feel tired. So what do? I take vitamins in the morning. My multivitamin has minerals and it has organic molecules called vitamins. And so these are cofactors and coenzymes. Notice a cofactor is inorganic ions such as zinc and magnesium and iron. And these help reactions to occur. They help the enzymes. Coenzymes are organic molecules such as NADH and FADH2. Remember those electron carriers that we talked about? And then there's prosthetic groups. These are also organic molecules that are non-amino acid atoms, and they too are also helping enzymes carrying out reactions. So enzymes do not work alone. There are other molecules that help them, and you intuitively know this because that's why we take multivitamins in the morning, or at least I do. Okay, so what other factors affect enzyme function? Well, there's two other factors, and these will be covered when we do lab, and we've already covered some of these: temperature and pH. So there's a sweet spot. It can't be too hot, can't be too cold, it has to be just right. The same thing with pH. It can't be too acidic or too basic, it has to be just right. But what does just right mean? Well, that's the tricky part. It depends on where you harvest your enzymes. If you harvest it from a human being, well, our temperature is at 98.6 Fahrenheit. So you would assume that most of our enzymes work best at 98.6. For pH, our blood is near neutral, right? So we would assume that most enzymes work best if you harvest them from a human being at a pH of 7. Now, if you harvest them from a fish that lives in the Arctic, well then maybe their enzymes work at a very low temperature. And what happens if this fish likes to be in an acidic pond? Then the enzyme prefers low temperatures and acidic conditions. So it just depends. So temperature affects the folding and movement of an enzyme, and pH also affects the enzyme shape and reactivity. So it can't be too hot or too cold. It can't be too acidic or too basic. It has to be just right. And if it isn't, it will affect the enzyme shape and its ability to function. So here we see two graphs, and I sort of love these graphs because it's showing bacteria. A bacteria, one that lives in a cool neutral environment and another one that lives in a hot and acidic environment. So if we look at the first graph on the left-hand side, we see that the x-axis is temperature in degrees Celsius. And when I think of 40 degrees Celsius, I sort of think of this is close to our body temperature. So notice here the sweet spot for the cool and neutral environment bacteria is near the 40-degree mark. But if the bacteria comes from a very hot and acidic environment, notice that its optimum temperature for enzyme activity is a little bit past 60 degrees Celsius. The same thing for pH. If it's in a neutral environment, then notice the blue line, it peaks. It peaks at near pH, maybe it's in between 6 and 7. But for the bacteria that's from a hot and acidic environment, notice that the optimum reaction or activity for an enzyme is maybe like a pH 3. So it depends on where you harvest the enzymes. But the interesting thing about these graphs, do you notice that all of these look like little mountains? So when we say that there's an optimum temperature or an optimum pH, notice that the graph always goes up and then down. It looks like a mountain. Not too little, not too much, just right. So most enzymes are regulated, like in class. When I want to get your attention during lab or lecture, whatever, I say, please, can I have your attention? Or when I see that you're talking, I'll say, please, can you stop talking until I myself finish lecturing? So I verbally tell you what to do, but how is it that we can tell an enzyme to start and to stop? How is it that we can regulate molecules? So there is something that molecules are regulated at the molecular level. And so the two ways of regulation are competitive inhibition and allosteric regulation. Competitive inhibition means that there's like a stopper at the active site. It acts like a plug. It won't let any more reactants fit. So when it acts like a plug, then the reaction stops. Allosteric regulation refers to a secondary site, a site other than the active site, and this will cause the enzyme to change shape. And in this case, it's like a light switch. You're using the same switch that goes on and off. Is you can either activate or deactivate an enzyme. Okay, so here I love figure 8.17 because at the top is competitive inhibition. So notice that the purple molecules act as a plug, and once they're there, they won't let the substrate fit into the active site. So end of story, the reaction ends. Now let's look at allosteric regulation. So if we look at the first part here, notice here we see reactants or the substrate given in the yellow molecules, but notice they don't really quite fit. But there's that secondary site, and a little yellow molecule the shape of a piece of pie fits into the secondary site. And now all of a sudden the molecules or the substrate can fit. This has turned on the enzyme. But notice in the second frame when we're looking at the blue molecules, which indicate the substrate, notice here they can fit into the site, but once a molecule fits into the secondary site, that little piece that looks like the molecule that looks like a piece of pie, once it fits into the site, notice that it doesn't fit in. So here it's turned it off. So allosteric regulation can either turn on or turn off the function of an enzyme. Okay, so regulating enzymes via covalent modifications. So this is true. Covalent modifications can actually affect the enzyme's primary structure. And some of this can be reversible or irreversible. It's good for you to hear about this. You won't have it on your upcoming exam, but it is good to know that it happens. So this is what actually initiates the activation or deactivation of a molecule. So notice here they show a switch off and on, and notice that there has been conformational change, and you can see it in the activation loop in orange. Okay, the next, we're near the end of the chapter, believe it or not, and I know it's been a long chapter, but this is, can enzymes work together in metabolic pathways? And the answer is, heck yes, they can. And it would be nice to think that most reactions are just single reactions, but in our body, there's a series of reactions to get work done. So in this example, we see A, B, C, and D, and notice that it takes 3 enzymes to go from A to product D. So this is a metabolic pathway. It is a series of reactions, and each of these is catalyzed by a different enzyme. Okay, so metabolic pathways are regulated also. So how do you regulate a series of reactions? You do it by feedback inhibition. So the very product at the very end of a reaction affects what's happening at the beginning. The product at the end affects an enzyme at the beginning of the reaction, and this is called feedback inhibition. So it says here, it feeds back to stop the reaction. So the abundance of a product will feed back to stop the reaction. And this reaction is going to connect to one of the first enzymes in the metabolic pathway. Figure 8.19 shows that you have product at the very end. It sort of looks like a little ninja disc, a pink ninja disc. But notice this pink ninja disc affects one of the first enzymes in the metabolic pathway. It looks like allosteric regulation, right? It fits into the site, and then the enzyme goes through this conformational change, and guess what? The substrate can't fit in there anymore. So this is what's stopping the metabolic pathway. Feedback inhibition. Okay, so how is it that metabolic pathways evolved? This will not be on your exam. I'd be happy if you know feedback inhibition, and that's about where things end for me. But the question is how is it that there were a series of reactions? Well, they call it retroevolution. In other words, it's a backward process. And so what you're doing is you're trying to evolve new substrates so that a reaction can occur. So what is it that I'm saying? So notice at the very top in step 1, you see one enzyme that controls one substrate and one product. But what happens if there's not enough of substrate 1? Then you have to produce substrate 1. So to produce it, it takes another reaction. So now you're working backwards to actually produce substrate 1 from substrate 2. Now you need more substrate 2. So another enzyme evolves to produce more substrate 2 from substrate 3. And this is called retroevolution. And this is how scientists believe metabolic pathways evolved. And this will be the end of chapter 8. Oh, excuse me, there's one other thing that you have to know, and I apologize. And you do have to know this. You have to know the difference between catabolic and anabolic reactions. So catabolic reactions, you break down molecules. You go from large molecules to small ones, and it releases energy. In anabolic reactions, you go from small molecules to big ones, and it requires energy. In other words, you're synthesizing large molecules, and it requires it. So a catabolic reaction is exergonic, an anabolic reaction is endergonic. And now we've hit the end of this chapter. Thank you so much, class.